The link between the gut microbiome and obesity has long been observed, but only recently has the scientific narrative shifted from correlation to causation. The gut is not just a passive digestive organ, it is a command center deeply intertwined with metabolism, hunger regulation, and immune balance. Increasingly, evidence suggests that disruptions in the microbiome, including the loss of key bacterial species or the malfunction of microbial sensing receptors like TLR5, toll-like receptor 5, may directly cause overeating and metabolic dysfunction. This is not merely an association, it is a mechanistic relationship, one that helps explain why diets and willpower alone often fail to correct weight gain in the presence of gut dysbiosis. TLR5 is a sentinel receptor found on the gut's epithelial cells, designed to detect flagellin, the protein that forms the tail-like structures bacteria use for movement. In a well-functioning system, TLR5 helps the body distinguish between benign and threatening bacteria, promoting immune vigilance without triggering chronic inflammation. But its role is not limited to immune detection. Research has shown that mice lacking TLR5 develop hallmark symptoms of metabolic syndrome, including increased appetite, insulin resistance, elevated blood sugar, and excess fat deposition, despite being fed the same number of calories as their normal counterparts. This strongly suggests that the loss of TLR5 function alone can rewire the body's hunger signals and energy storage processes, independent of diet. One proposed mechanism involves TLR5's influence on gut-brain signaling. When TLR5 function is lost, the balance of microbial communities shifts, often toward motile, inflammatory strains that produce abnormal levels of flagellin and other microbial byproducts. In the absence of a properly functioning receptor, the gut may become blind to certain microbial cues, triggering systemic inflammation and disrupting the normal signaling between the gut's neuropod cells and the brain. As a result, satiety signaling may be blunted, while reward pathways in the brain become hyperactive, leading to increased food intake and cravings for calorie-dense, highly palatable foods. Beyond TLR5, other disruptions to microbial diversity and structure show similar effects. Studies in humans have consistently found that individuals with obesity tend to have a lower diversity of gut bacteria, with reduced populations of beneficial strains such as Akkermansia mucinifila, Faecalibacterium prosnitziae, and Bifidobacterium. These microbes are critical not only for fermenting dietary fiber into short-chain fatty acids, SCFAs, which regulate hunger hormones like GLP-1 and PYY, but also for maintaining gut barrier integrity and keeping inflammatory signals in check. When these beneficial bacteria are lost, often due to poor diet, stress, antibiotics, or early life disruptions, the gut becomes more permeable, more inflamed, and less able to communicate effectively with the brain. This breakdown in communication creates a dangerous loop. Inflammation and dysbiosis dampen satiety cues, while also altering dopamine and serotonin signaling in the brain, which regulates motivation, mood, and reward. The result is a biological environment that favors overeating, not out of choice or lack of discipline, but because the signals that tell the brain to stop eating have been physically altered or suppressed. Meanwhile, fat storage mechanisms become more active and insulin resistance sets in, locking the body into a state of caloric overconsumption and inefficient energy use. Adding further weight to this theory are fecal transplant experiments in which gut microbes from obese individuals are transferred into germ-free mice. The mice rapidly gain weight and develop insulin resistance, despite no change in their food intake. In contrast, transplants from lean individuals can confer protective effects, even improving glucose tolerance and reducing fat accumulation in animals predisposed to obesity. These results demonstrate that the microbiome itself can drive or reverse metabolic states, acting as a switch that controls whether the body operates in a state of balance or excess. If this microbial dysfunction is causative, not just symptomatic, it means that reversing obesity may require more than calorie control or exercise. It suggests a need to rebuild microbial ecosystems, restore receptor function like TLR5 signaling, and reestablish the gut's ability to accurately detect and relay metabolic signals to the brain. This could be accomplished through a combination of dietary interventions, high in fiber, polyphenol, and fermented foods, prebiotic supplementation, carefully selected probiotics, or even next-generation therapies like synbiotics and microbiota-targeted pharmaceuticals. Ultimately, understanding obesity as a microbiome-driven disorder redefines the condition from one of personal failure to one of biological miscommunication. The body is not broken, it's just receiving distorted messages from a gut ecosystem that has been compromised. By healing that ecosystem and restoring the sensors that regulate appetite and metabolism, we may finally move toward more effective, compassionate, and lasting solutions for one of the most widespread health challenges of our time.